Okay, we see a sea cucumber, a holothurian. I can see
It's
that.
going to be number 2, but I think we have the diagram. You don't have a copy of the diagram? Oh, okay. So it's going to be like 936 slash 2.
Okay, so stand by. We're setting up for the transect.
Not slash, hyphen. Dash. Yeah, no, not on this one. Just like
Yeah, that's a different checklist, but
936.
up there.
And then you start, and then they will flash the lights each 100 meters. But when we finish a total of 500, they will be the end of the dive. So, sorry, the end of the transect. So, at that moment, you also record this. They're planning. They're planning their transects. So no, not yet, because they're all still like putting the waypoint. So yeah, for the transects you use this one. So here you type this like 936 transect 2 start. No, not needed. And then at the end, they will flash the lights.
Double flash of the lights when you're ready to start, and then if you can just flash them every 100 meters, please.
So once you see they flash the lights, you can.
Great job. Good, right? Yeah, we're gonna be seeing it and stuff, so you can follow. Always, okay.
Okay, here we go. So as I mentioned before, we're sitting in a room on the ship, nice and comfortable, and we're able to observe all around us on the seafloor, all around the ROV Sebastian, because there are cameras that are positioned several places around the ROV. So what you're seeing is just one part of the live stream, one of the cameras. Sometimes we switch out. And so if you were sitting in the room with us right now, you would see all kinds of monitors, literally on 3 walls of the room. And they're displaying different views from the different cameras, as well as any of the data related to the dive, the heading that we're going, the depth at which we are. We are around 962, 63 feet Yeah, sorry, meters, 963 meters. I'm American, sorry. Um, and let me see, we get all kinds of data, temperature, heading, we have sonar, so we're able to see what's ahead of us. I'm sitting up in the front row, so I have one of the front row seats, the expensive seats, along with the ROV team. And then behind me are members of our science team. So, and every now and then some of the crew come in and they're real interested in what we're doing as well. We have monitors all over the ship, even in our staterooms and our cabins, so we can see what's going on at any point during the dive, which is really great, because I can be in another room on the ship and I can see something that it'll most likely be a sponge that I would like to get a closer look at, or maybe even collect, and within just a couple minutes I can walk slowly from where I am and come to the ROV room, mission control, we call it mission control, and be able to, so anybody on the ship can participate at any point in time, just like you're participating, watching on your monitor at home. One of the many advantages of remotely operated vehicles, which by the way it's a submersible. It's tethered to the surface, so it's not free swimming. It's tethered to the surface, so through that long tether, now remember we're 961 meters deep, so it's a very long tether, and that all the power to the submersible is going down through that tether. And the communications that, this excellent video that we're seeing and all the other communications, the telemetry is all being transmitted back up through that tether. I've been real lucky in my career. I've had the opportunity to go into manned submersibles. I guess the politically correct term is human occupied vehicles, but manned submersibles. So I was actually sitting in the submersible and being able to kind of do pretty much like this, observe what was going on around me, be able to document using video and collect. So the basic, well, there are a lot of differences. So the human occupied vehicle runs on batteries. So after a few hours, you have to come back up to the surface and recharge the batteries and then go back down again. And depending on what submersible you're in, batteries may last longer, depending on what kind of work you're doing. If you're doing a long transect like this, the batteries run out. Here, with the ROV, I mean, we're on a 14-hour dive, so we probably, would have had to come up a couple times in a manned submersible. One of the other differences is that in a manned submersible, it's just the people who are in the sub who are able to see what's going on around you. I know at one point, I think it was James Cameron, the producer director of the Titanic and the Avatar movies, tried doing a fiber optic transmission through the water, but it was really difficult. With a remotely operated vehicle, we can transmit what we're seeing and share this exploration virtually unlimited around the world. So that's how you're able to share in this exploration with us, and that makes it so much richer for us. And so when I was in the manned submersible, I'd be sitting usually next to sitting with a pilot, and then and there'd be a scientist and another submersible technician in the back. And it was only the expertise that we had that we could say, okay, well, I think this is a, octocoral, this is a tunicate, forget about fish, I don't know fish at all. The advantage of being in a mission control with a number of scientists with different expertise is I can look around a room and I can say, hey, Bia, what do you think this is? Is it a, isopod or, what is it? And so we have the collective expertise and multidisciplinary, so we have a lot of people who can contribute. Flash. Ready to flash? Okay, flash again. Okay, great. Thank you. Okay, Thea, I promise to pay more attention instead of
You're doing
talking
great.
the whole time. Okay. We've seen a lot of sargassum on these dives. It's dropped down, sedimented down from the surface. Those of you who live near the coastline in the Caribbean know that we have these huge mats of sargassum that are floating ashore. And I think yesterday we showed you a video of sargassum, just outside the, just outside the ship. And so it's, it's not unusual that we're seeing the sargassum on the bottom. And that breaks down over time. Looks like a coral that's been knocked over. Keep that. I was watching as the ROV descended this morning. There were a lot of really beautiful midwater organisms during the descent. So imagine it'll be equally beautiful when the ROV ascends later on. And let's see, seconds, probably another 5 hours, yeah, 4 hours, 5 hours left. Okay, so I don't have to keep counting these squirrels.
Where are you? I mean, that sounds way to you.
That's 200 inches.
Okay. Just chomped off another 100 meters. So it might look like there's not much here, but there's a lot of activity going on underneath that sediment and that mud. You can see occasional mounds, and right before I came on, I saw a really big burrow with 2 large holes on either end, and then a big mound of sediment that had been cleared out. So there are all kinds of worms, polychaetes, gastropods, you name it, living in the mud. And then of course the sea cucumbers are crawling across the bottom, and they're eating that mud that contains all kinds of organic matter, bacteria, other microorganisms, and then they're processing that through their guts. And sometimes you'll see long fecal pellets so you'll see tracks, fecal pellets, mounds. All of that is evidence of life beneath the surface of the sediment. There's a sea cucumber. Another shrimp. Such a pretty color. Some of the other activities that are going on are plankton tows. So at night, we're doing tows for phytoplankton and zooplankton and ichthyoplankton, so looking for fish larvae. Look at that. Is that a little star? No, what is that? So there's something going on 24 hours a day out here during this expedition. Sea urchin. We got one of those sea urchins yesterday, and they're very delicate. You have to be very careful when you pick them up. They're also very spiny, and those spines are quite nasty if you get stung by one of them. Ready? Okay, great. Reached our next waypoint on our transect. We've now gone 300, I think, of the 500-meter transect. Corner, we can see a hexactinellid sponge, hyalonema. Hyalo means glass, and nema means thread. So this is glass sponge with these long spicule threads. There's another one right in front of us, hyalonema, and this one has some brittle stars on the stalk and zoanthid, so it looks like it's waving. Oh wait, those is that a rock? Yeah, lobster. Yep, spot lobster waving to us. Pretty anemone. Surrounded by sargassum that's sedimented down from the surface. another anemone. And sea urchin, shrimp. So you all know by now that every hour we switch out, so you don't have to listen to the same person all day. And I'm getting ready to switch out, I guess right now, to my colleague, Dr. Megan Conkling, and she'll be your host for this next hour, finish out the transect and do some more observation and collection. Thanks for joining us. And by the way, I just wanted to thank the ROV crew again. They're really talented, and we're very fortunate to have this crew here, really making these dives possible for us.
Good job.
Thank you.
I'm great. How are you? Hello, everybody online. My name is Megan Conkling, just like Shirley had mentioned, and I'm going to be narrating this section of the live stream for you all. So my role on this ship is a co-PI, so I'm going to be helping Chief Scientist Steve Amon with exploring the deep sea of Trinidad and Tobago, and my main role is to train the people of Trinidad, and specifically the UWI Museum, on how to cryopreserve marine benthic invertebrates. So that is going to be a training portion that we're going to be working on this vessel. So I'm going to be taking advantage of a lot of the examples that we're collecting using the ROV. We're seeing a lot of different types of fish in this area.
400 meters transect.
So we do transects on these ROV dives in order to better document the habitat and the organisms that are living in this area, so that we can do more analyses to better understand the potential of the waters and the surrounding habitats in the area. So we take specific images based off of the distance of the ROV off of the bottom that allows us to better understand and quantify what we're seeing and how to document it properly. So this transect and the information that we'll collect with it will help us to guide the government with potential marine protected areas and just help them, advise them on the habitats that are in their backyard. We have a sea star on the bottom, as well as another shrimp. Looks like we have another sea cucumber ahead. And maybe a sea pen off of to the side. Another sea cucumber. Transect is complete.
Heading south.
Continue heading south. Continue heading south.
Yeah.
Okay. Yes. Continue heading south.
Do you see something or am I correct?
Or close up. Okay. Sounds good. Another sea pen on the floor. So my expertise is in sponge cell culture, but like I said, while on the boat, I'm going to be training the museum curator to cryopreserve benthic marine invertebrates. And this will allow for Trinidad to be able to develop their own living biobank. So with the animals that we collect or creatures that we collect with the ROV, we are going to be able to cryopreserve them in a cryoprotectant, and this media that we're going to put them in allows us to freeze them down to negative 20 degrees Celsius, or even colder, down to negative 80 or negative 100 degrees Celsius, in order to preserve the living tissue or cells of these organisms. Look at this little fish here. Oh my goodness. Look at the coloring on that. We have another sea urchin there on the bottom. And looks like another Venus flytrap anemone. Beautiful. Oh, what's that sitting on the bottom there? Can we get a shot of that? Yeah.
It's a sea robin.
Oh, okay. It looks to be a sea robin. Yeah.
They have like this
Little arms. Have we seen one yet?
Yes, yesterday we
Okay.
saw one, but in the background, this is the first one that we are able to
Nice.
close up.
Look at those eyes.
Maybe cheaper contrast stuff.
Yeah, so it's armored sea robin.
Okay. All right. It's an armored sea robin. So just a reminder to everyone on live stream, we're using a remotely operated vehicle, which means that we are still tethered to the vessel. So they have to be well coordinated in order for us to be able to stay on track with everything. So, and they're doing a fantastic job. So we appreciate all of their hard work.
That's our Arabian rules.
More more sargassum clusters. Looks like we have another Venus flytrap anemone with a squat lobster with it. He's waving to the cameras. Sea cucumber with another Venus flytrap and squat lobster. Looks like a sea pen, as well as some of the common fish that we're seeing, the halosaur. Another sea cucumber. another stocked sponge, also known as a halanema. We have a sea star here. Allosaur fish. So this expedition has brought together a lot of different people from a lot of different fields. So geophysics, biology, plankton specialists, as well as people that look at macro and microfauna. It's pretty extensive on this vessel, the expertise that we have, and we are all very excited to be learning from each other, because no matter how much schooling you do, there's always more to learn, especially from your colleagues and collaborators. And this has been a wonderful experience thus far, and it's only day 2. We got it's another sea cucumber and glass stalked sponge, a couple of them. Oh, got a nice sized shrimp here with a halosaur fish. And glass sponge with a squat lobster in the back. And there's some zoanthids on the stalk of that sponge. So they're taking the opportunity to utilize that space. Otherwise, they would not be able to settle on that soft sediment that they have on the bottom here. So they've taken the opportunity to utilize that prime real estate that the sponge only uses in order to get up and out of the sediment and into the water column to filter better. And of course, we have the beautiful squat lobster, who's just hanging out. Oh, we got another tripod fish. Can we get a close-up of that? A shrimp and another sea urchin in the background there.
Good job, Dave. Can we pull some movements? Great job, Dave. Can we pull some movement? Oh, good. Great job, Dave.
Yeah, right.
Hi, mate. Can we pull some movement, please?
All the way.
Like I said before, there's a lot of coordination that's involved between the ROV pilots and the vessel in order, because we are tethered to the vessel, there's a lot of coordination that's necessary in order to keep from getting the ROV pulled off and making sure that the ROV is working properly and safely maneuvered. So we appreciate all the hard work that's involved with allowing us to do these dives and collecting the data and imagery that we get from these experiences. We can continue on.
Continue to move.
We'll find another one. Got a sea cucumber and a stalked glass sponge, the Halanema. I have another one of the large shrimps there. Just like a ctenophore in the water column there. Beautiful. You can see the light show going on with the ctenophore there. Beautiful. Got a sea star down at the bottom. And another halosaur fish. And maybe a small anemone as well. A sea urchin. There's a glass sponge there at the Halanema. Looks like we've got more zoanthids, a squaw lobster, and an anemone on it. Big shrimp there on the left.
Perfect.
that a little anemone? Potentially? Another sea urchin. Sea star over there. Looks like he's missing a couple legs. Another sea pen there, and the allosaur fish. Looks like these halosaur fish are pretty common in this area. Another brittle star. You got 15 minutes.
Okay. Five, six. There we go. Or you can ask them to put something aside.
Heavy,
Okay,
right?
put something aside. I see. Go, problem.
So this 20 just interested to get from the sea that after the push cars done.
The reason why you get that dust whenever you stop chewing is so low. So this dust is continuously falling here. Like you're sucking it. So when you stop, dust comes. Sometimes
Trash?
The trash?
Trash? If it is, we should log it. Trash? Can't tell. Ooh.
Got another sea urchin on the bottom. More sea cucumbers as well. Lots of little shrimps in this area as well.
I'm ready, gentlemen.
You ready?
Looks like a couple sea anemones on either the remnants of a stalk from a sponge. More sea cucumbers.
Can we zoom in?
Hmm?
Zoom in.
Can you zoom in more on that? Mm-hmm. Thank you.
Yeah, it's an anemone. We'll never get it.
Looks like another anemone.
Oh.
Yeah, continue to go. Thank you. Okay. Can you flip the camera up a smidge so we don't see the cable tie on the bottom right? Thank you. Looks like another anemone on the sand there on the left-hand side. We have a brittle star and a small sea urchin, looks like. Looks like this one's lost a couple of legs as well. Oh, we have a ctenophore in the water column. Can we get a closer zoom on that in the bottom? Yeah, on the
Yeah, it's
benthic.
more like a tree.
Okay. Yeah.
Oh, crinoid.
Looks to be a crinoid.
Yeah. Beautiful. Let's get some pictures.
Okay. We'll stay here to get some pictures if that's all right.
The brittles there on the bottom.
Yeah, tiny,
They kind of do.
tiny brittle
Kind of
star there at the
unrolled.
bottom of the stalk.
We need to pick it up.
Perfect.
Reflection?
All right. Yeah, we're gonna try and collect this.
Like this.
Suction.
Oh,
Suction.
suction.
Yeah,
I'm gonna need to grab
the star. Okay.
Tree, Jeremy. You can do a stop one.
We're gonna try and use the arm to collect. Yeah, we can do coral quiver one.
For a blue.
We're gonna try to place this in coral quiver one.
Multiple.
It's we.
Perfect. Wonderful. If we can get some close-up shots with it in the arm, that would be great. Beautiful. We'll see when it gets to the top if we still have that little brittle star attached or not.
Look at all that.
Beautiful picture there. Perfect.
So that's the kind of.
Thank you, yeah. And coral quiver one if we can.
Yeah, they're probably, well, they're not sort of cry knots, but they're sort of good at crustaceans. We'll see.
Oh, I think we just lost the brittle star.
Oh, it's true.
Yep,
Fallen
he's fallen back to the bottom. He lives another day. Perfect. All right, well, that's the end of my shift, so I'm going to pass off the mic to Eugenia.
Collected.
How is everyone doing? So that was my lovely colleague Megan narrating. So I'm Eugenia Thomas. I'm a marine biologist with dual specialty in deep sea biology and tropical marine ecology. I'm also an MPhil candidate at UE studying deep sea woodfalls and deep sea debris in the Caribbean. And so what you're seeing is that we just collected a crinoid, and this belongs to a family of echinoderms. These are 5-star symmetry organisms. And what we also saw was a brittle star, which is another type of echinoderm found colonizing on the crinoid, also known as a feather duster worm.
You're a biologist.
And so today's site, we are due west of Tobago.
Sorry, the razor?
Yeah.
I'll put it on.
So we are exploring the west of Tobago, and so far we've collected many different species. We have collected sea urchin, sea cucumber, and we just collected a crinoid as well.
Yes?
Yeah.
Will do.
And so, as my colleagues will have mentioned this morning, part of our objectives of our expedition is building deep sea capacity, regional and locally, and also building a biodiversity database of species found in the seafloor of Trinidad and Tobago. And again, this expedition is very, very important for us here in Trinidad and Tobago, as this is our first locally led Tobagoan expedition. Of these 25 scientists, 16 of us are Trinbagonians, and this is a very monumental occasion for us. And so coming up on our frame is a sea cucumber, another animal of the echinoderm family. And as mentioned, these animals are 5-star symmetry. And if you look really closely on the sediment seafloor, you also see that there are tracks behind the sea cucumber. And these tracks that you may have seen throughout the dive are actually evidence that there are life within the deep sea. So even though you don't see an animal present, you will see the tracks, and it just goes to show you how diverse the deep sea environment is. You can see a small species of fish that's moving along throughout the water column. And throughout our dive, we've seen a lot of sargassum on the seafloor as well. And as you know, there are many sargassum throughout the Caribbean, and these make its way into the seafloor, and they actually provide organic matter to animals living within the sediment. And so they play a very good and important role in the deep sea environment. And here's another sea cucumber, and we also just passed a small sea anemone.
Could you resume the move?
Sure, yep, I'll get going now. Thank you.
Thank you.
If you zoom in on that, super, I think. Yeah. So what you see here is a fecal cast of a sea cucumber. And so if you look real closely on the sediment, you also see tracks. And this also shows you the route in which the sea cucumber take. And you also see a brittle star and a small soft coral. And so again, these fecal casts shows you that a sea cucumber has been around in this particular area. And this is one of the many biological evidence that exists on the deep sea floor. So you can see tracks, and then you can also see fecal casts like this one in the frame.
Right?
Thanks. I'm sure you also familiar with the shrimp as well. We see a lot of these species on our dives.
Listen, yeah, it's gonna be here. So you're doing great.
And here's another sea cucumber. And these animals also play an important role in nutrient cycling. So the important role in the environment is bioturbation. So they basically help cycle organic matter on the seafloor and also helps in nutrient cycling for the deep sea environment. So what you're seeing here in this frame is another crinoid. So we would have collected a sample like this earlier. So again, a crinoid is another form of echinoderm. There are actually 5 animals within the echinoderm family. There are sea cucumbers, feather stars, also known as crinoids, starfish, brittle star, and sea cucumber. And all these animals share one thing in common: they are 5-star symmetry. Yeah, it's okay. So crinoids are filter feeders, and we're also coming up on another family of the echinoderm, a sea urchin. And there's another crinoid. So again, these animals are filter feeders, and so they feed on microscopic organisms or organic matter in the environment. And if you look really closely in your frame, there's also a small comb jellyfish that just passed us. And you can also see another fecal cast of a sea cucumber. Again, even though you don't see the sea cucumber, this fecal cast shows you that a sea cucumber was once here. And you also see the tracks near to the fecal cast as well. And here is another one of our favorites, a tripod fish. We've seen a lot of this on the dives earlier today as well.
Them have the same thing, like the white.
Yeah.
There's white thing hanging. Yeah, like a person.
Oh.
The other one that we found had the same.
Yeah.
I mean,
So it's just resting there looking, all wonderful in this glory.
Cool.
So in the frame as well, you can also see another species of fish. And so again, we are exploring to observe the different animals that exist in our own backyard, and also to see the type of behavior that they would have shown as well. And here's another crinoid. And so all these on the sedimented seafloor, you also see some holes, and those are burrows in which animals such as crustaceans will have buried itself and feed on the organic matter within the sediment. And so even though you're seeing even though we are on the sedimented seafloor and we are not seeing any physical animals, there are animals that are buried in the sediment. And so in this frame, you can also see a soft coral, and one of the favorite animals that attach itself to this type of coral are spot lobsters. And so these soft corals, their structure provide a home or a habitat to animals like spot lobsters or brittle stars, sea anemones, etc. And so this sargassum you see here, it would have fallen from the surface and wrapped itself around the soft coral. And you can also see some brittle stars on the sediment as well. You also see some pieces of shells as well. And again, these shells and the sargassum provides nutrients within the sediment in which animals can feed on. And here you see a crab really moving along the seafloor. And so this is another species that will also be attaching itself to the soft coral.
It's a hermit crab. Yeah, it's a sign.
So in the frame you can also see a hermit crab and a snail. Very, very beautiful. And in this frame you can also see another snail as well. Oh my god, this is beautiful.
Purple.
Yeah. So you see it looks purple, green, you know. Oh, and it's gone. It'll be back. Oh, there it is. You also see another hermit crab in the frame as well. So it's just resting on the seafloor. And these organisms also feed on the organic matter within the sediment as well. As well as other small organic matters within the water column.
Love it. It's 3D printed, this is like
Yeah. And so if you look at the shape of this, it's a spiral shape. Whereas the hermit crab have sort of a conical shaped shell. Beautiful color. Yeah.
like it.
Yeah.
Like it.
Yeah, it looks nice. And in this frame here, you can see a soft coral. And there's many other organisms on the structure as well. Yeah. Yeah.
Así es como que colectamos.
You see sea cucumber, soft coral as well.
think that's actually a mushroom. It's an armored sea robin. Armored sea robin? Yeah. Sea robin.
And so this beautiful species of fish here is an armored sea robin. I mean, just look at the structure and the colors. Beautiful. And there's another sea star in the frame as well. Zooming on that kind of long cylinder.
That was a wood pole. Remember how it's a wood again? Yeah. Maybe a wood. What do you think? Seems like wood, no?
Look like a little plastic.
Yeah. Two.
And so there's a small piece of trash next to the soft coral. Looks like a bottle cap. Small container. And so our activities on land affect deep sea ecosystems. And so debris items like fish necks also become entangled in corals or sponges. And these can also affect the habitat structure that these organisms provide.
Let's wait here for a minute. Wait here.
And so there's a lot of small critters that is moving around this frame as well. You can see some polychaetes, also known as worms. Some reminiscence of sargassum. And you also see some broken shells as well.
Sample this one. I think it's a good size for a caterpillar, right?
Yes.
Sizes. These are all empty, except for this one. So you can put it here.
Can you put it in too?
On the second part,
Yeah.
no problem.
I don't know small frame grips.
Are we using arms for that?
Sorry?
Are we using arms for it to pick it up? Are we using the arms to pick it up?
Es cuando lo tengo en el brazo y va a ser que la cámara no zoom ahí sí se lo tengo en sample. Todavía.
if that helps. It's in the wood section. It moves. You're joking. No.
Yeah, it's good.
Escape.
Okay. For her.
Really?
Oh, that's
See,
Felt
that's
it.
why.
They moved it. Oh man.
Yeah, una vez que
Never seen it. I've never seen this happen. I was not aware that.
You can find another one.
All right. Oh. Name. We'll try.
That's fine. I think it might be because we're seeing water.
What was the special about this one?
I don't think we ever saw them.
Did it look colder?
Yeah, this one look a bit different. Wow, so here you have a trash bag entangle itself on a sponge colony. And so this is exactly how our activities on land impact these organisms. Additionally, you also have organisms such as sea anemone and or brittle stars that would have been entrapped under the plastic bag, causing immobility, and sessile organisms like these need to be able to move in order to find food. And so one of our objectives on this expedition is to collect eDNA. So sponges are filter feeders, meaning that they feed on organic matter and microscopic organisms within the water column. What also happens is that they also absorb DNA from other organisms as well. And so by extracting this DNA or environmental DNA, eDNA, we are now able to decipher the many different organisms that exist on our seafloor. And so sponges serve as an intercept between investigating species biodiversity in the deep sea environment. And the frame you can also see another one of our favorite crinoid. There's another snail on the seafloor as well. So here's a soft coral, and you also see gooseneck barnacles attaching itself on the structure.
Yeah. True. I mean,
Like a
I
branch.
don't think so. Like a branch or something.
So again, many of these holes that you see on the sediment are animals that would have bored itself to feed on or escape from predators. And you've seen a lot of these fish species as well.
What's the urchin?
And so in the last frame, you see a sea urchin, and there are other two organisms that exist within the Echinoderm family, the sea star and the crinoid. Here's another type of fish. This one's a bit different from the last one we saw. Wow, so here you have many different organisms within this frame. You have a shrimp, you have a soft coral, and on the soft coral you can also see a sea anemone, squat lobster, and gooseneck barnacles, as well as sargassum. And if you look real close, you can also see a brittle star on the sediment as well, and a hermit crab.
It's actually a dead sponge or a dead stalk from another animal, and then we have like a lot of swans probably that are like hiding right now their tentacles, and some barnacles. You can see like some of them are open and most of them are closed, so they retract.
So this is actually a dead sponge. Still provides a home for many different species as well. Here's a hermit crab. So it appears as if it just came out of the burrow that it was in, and now it's moving along its journey. Yeah. So many organisms within this frame. And again, these sargassum and these species of shells that you see on the seafloor provide organic matter, which are then used by other organisms such as sea cucumbers. And so this overall assists with the biological pump within deep sea environments.
Muito similar to the one we yesterday, right?
No, I think this might be the one before.
Is the one from yesterday? No, from the one from the
No?
Yeah.
Yep.
Go ahead.
It's not opening now, it's a drop.
Okay, there's one more.
So there's so many tracks on the seafloor as well, and this is evidence that they are alive. Yeah. So in this frame you have a Venus flytrap. Some sea anemones attach itself on its stalk, as well as our another favorite, squat lobster. And you also see some brittle stars in the sediment as well. And there's also a small species of crab at the end of the Venus flytrap as well.
Yeah.
Yeah. And you also see some of our beautiful snails as well. There's actually another spot lobster.
Good?
Yeah. Yeah. Then you can also see some sea anemones at the bottom as well. And so these findings from this expedition will help guide or provide recommendation to policymakers, which in turn will better protect and conserve our deep sea environment. And in the frame, you can also see a comb jellyfish. So these organisms are filter feeders. And we're also coming up on another fecal cast made by a sea cucumber. And again, this provides evidence that this animal was once in this area. And in the frame, you can also see another comb jellyfish. And these organisms produce their own light, also known as bioluminescence. Wow. So this particular jellyfish is known as a nattophore. Mm-hmm. Yeah. And so sometimes a lot of these small depressions you're seeing on the sediment are from animals that will have rested on the seafloor. And similarly, the holes in the sediment as well are from animals such as crustaceans, like hermit crabs, that will have burrowed itself within the sediment to escape predators or to feed on the organic matter created by sargassum or marine snow. And so some of these small white objects that you see on the seafloor are petripod shells, and they also provide organic matter to deep sea sediments. Okay, so that's the end of my narration, and I would also like to pass you over to my colleague Zahi, which will be now leading this dive. So thank you very much.
Hi everyone, good evening, good morning, depending on where you're coming from. My name is Zahir Hussein. I'm the curator of the UE Zoology Museum, one of the contributing entities to this expedition. Happy to be here with you as we continue to explore the deep. So we're continuing our transect. We're into the last couple hours of this dive, continuing to observe and document. Couple Holothuroidea, some sea cucumbers. We often find sea cucumbers in our shallow coral reefs as well. See quite a few of my students online. Hello everybody. And we also have a deep water shrimp in midwater here in the water column. Beautiful red color. One of our deep sea fishes, possibly Aldrovandia, but that has to be confirmed. Very large eyes, as expected for an organism that lives so deep down with so very little light. So a lot of the energy that powers this system down here comes from marine snow, which originates up at the surface. But most of the energy comes from chemosynthesis, from thermal vents, where chemosynthetic bacteria convert hydrogen sulfide into sugars that help to power the food chain down at the deep. Sea star, and something running along the surface, running along the benthos. It's a little crab. So we're gonna zoom up and see if we can get some pictures of this guy. Oh, he doesn't agree. So they're not not accustomed to a lot of light, so sometimes the light can elicit this sort of behavior. He's coming back. It's a great shot there. And we let him go on his way. Ooh. I think that fish wants to be on camera as well. We sometimes see bioluminescence even with the lights on. We keep the lights on because we're currently doing a survey of the bottom, so we need to see everything. But we often see bioluminescence in many of these deep sea creatures. A small anecdote, we got one of the worms out of our sample yesterday, and he was still alive when we were preserving it. We actually saw a bioluminescence in the lab from him. So that was pretty cool. So as we go along, every creature that we see along our transect is being recorded. Because we don't know much about our deep sea, we're trying to create a baseline. And one of the ways that you do that is by doing abundance and density surveys. So what we're doing now is trying to determine the density of organisms that we see along our transect. Many of you would have done this in your own research and in your own schools, where we looked at number of individuals per given area. It's the same thing we're doing here, just on a little bit larger scale. Oh, that's a interesting jelly. We're gonna try to get a good shot of him. Because we're so close to the seafloor, the props sometimes stir up the bentos a little bit. But we have some of the best ROV pilots in the game. So as you'll see in a moment, we're gonna get a good shot of this guy. A little bit of bioluminescence there. For those of us who wanted to see bioluminescence, there you go. And every minute of the dive is actually being recorded both on video and on a log. Beautiful jelly. So all of the samples that this expedition yields are going to go back to Trinidad and be stored at the UE Zoology Museum and at the museum at the IMA. Some of the samples will be sent to the States and to Canada, where colleagues will continue their own research on them. But it must be noted that all copies will be housed at the UEZM and at the IMA's museum, which is a great stride forward for us in Trinidad, because we have had expeditions in the past, but most of the samples, or very little of the samples and specimens, actually stayed in country. This is the first dedicated expedition where the specimens will be housed in country, both whole specimens as well as genetic samples for further research. So some of my volunteers at the UEZM are going to have a lot of work coming up. As we try to document everything that comes out of the expedition. We got something in front of our camera here. Can isolate it. Seems to be a lot of turbulence down there right now. Many of the organisms that you see in front of you are being attracted to the light. Coming up on another soft coral, I think. Is it? Yeah, another octocoral. Yeah, keep moving. We're okay. Octocorals, not to be confused with their cousins hexacorals that dominate our shallow coral reefs, particularly our scleractinian corals that form the very dense, thick reef framework that we associate with coral reefs. And when we hear coral reefs, we think of this large expanse that you have in Tobago or maybe even in the Great Barrier Reef, the Mesoamerican Barrier Reef. But there are other types of corals like our octocorals that are soft, found at depth or in high latitude settings. Another interesting jelly. Moon jelly possibly, bioluminescence. Jellies are actually related to corals. Beautiful. Yeah. So it is a bit choppy down there. I'm not sure choppy is the right word, but a lot of turbulence, a lot of current. Some of you may be seeing two green lights on your screen. Those are projections of our the laser set to 10 centimeters, I believe. So no matter how far the ROV is away from the organism, the lasers change and maintain 10 centimeters. It's very effective in helping us to measure the size of these organisms. Oftentimes we don't want to take some things, but we want to get as much information as we can. So size is one of the morphometric characteristics that we always tend to take note of. Crustacean? Many of these creatures are new to me and to many of us. We have some structures, looks like plastic bags and a possible tree fall. Maybe, but certainly plastic bags. We have some crabs, some squat lobsters. Organisms tend to like structure and they congregate around structure, as we're seeing here. Some sargassum as well. Yeah. Yeah, go ahead. Keep them. Oh, that's a lovely shot there. Yep, so even at 1,000 meters, we still have anthropogenic inputs. Yeah, many organisms would utilize these woodfalls as habitat. Or octocorals. Nice little shrimp. I'm sure I'm not alone in thinking, I wonder what that tastes like. Another possible Aldrovandia. There he is. He seems to want to be on camera. An amazing shot, despite all of the current and turbulence. Sea star and octocoral. That shirt. An anemone, and we have a squat lobster. We keep seeing squat lobsters on sponges and anemones, and seems to be somewhat of a symbiotic relationship. We also see anemones, squat lobsters, and glass sponges together quite frequently, possibly holobionts. Holobionts are a group of organisms working together. I'm sure we know what symbiotes are, but holobionts are more than two species. So another example could be our hard corals. Hard corals have a relationship with the photosynthetic zooanthellae, but they also have a relationship with the bacterial element that surrounds them, and together they are called holobionts. And it seems to be that this gives them a bit of a selective advantage. We've also seen examples of other crustaceans, like our squat lobster here, guarding corals. Crabs of different sizes protect the corals from corallivores, and they in turn receive habitat from the corals. So closely evolving together and having a beneficial relationship. Oh, beautiful. Wow, the skill of our pilots is amazing. They have to anticipate the movement of the organism as well as recognizing that there's very little resistance in the water. If you push one way, it's going to go that way. So they have to think in advance about which direction they're going. They're awesome. And for those of you who are wondering, the room that we're in, Command Center, it kind of looks like Star Trek. Really, really cool. And welcome to all visitors from all around the world. Happy to have you. We have some structure. But please remember, we are on a timed mission, and we do have a certain distance to cover between our waypoints. So we can't stay too long in any one position. That's why you see us moving along. But things are still happening. Every organism we see is currently being logged by my colleague Giuliano, who's on the logging station right now. Beautiful jelly. Some of you may ask why we're doing this. Well, we're doing this because so very little is known about our deep sea. I mean, if you think about it, we know more about space than we do about our deep sea. We've explored more of space than we have of our own deep sea on our own planet, especially in our small island developing states where we don't have the capacity really to do these sorts of things. We welcome the Schmidt Oceanographic Institute and their help, as well as species and all our other partners who have worked very hard to bring this beautiful ship Falcor 2 to Trinidad's waters. I just want to shout out the University of the West Indies, the Institute of Marine Affairs, most importantly, our lead institution, Species, which is a non-profit NGO in Trinidad and Tobago, that undertakes scientific research, education, and advocacy. We also want to shout out the Harbor Branch Oceanographic Institute, Florida Atlantic University, the University of Calgary up in Canada, so they've come down to get some warm weather, Dalhousie University, Canada, the National Museum of Natural Sciences, Spain. Other supporting institutions: Ocean Discovery League, USA, Pure Ocean, France, the Explorers Club, lovely name, the Explorers Club, USA, the Pew Charitable Trusts, USA, and the Anthony Ed Sabga Awards for Caribbean Excellence. Those are our supporting institutions and our participating institutions. And a big shout out goes to them. Without their help and their vision, this expedition would not have come about. So great thanks to all of you. Some sargassum and a octocoral. So as we proceed on our survey, I just want to fill those of you who may not be aware on our project objectives. Our approach is novel and holistic, encompassing science, policy and communication, as well as our own personal goals. Specifically, we would like, and we are, doing high-resolution mapping of the seafloor. Some of our colleagues from Canada are doing heat flow data, obtaining heat flow data along previously acquired seismic transects, so you realize we're sticking on very specific zones to elucidate patterns of seafloor fluid discharge and implications for biodiversity. So what my colleague was telling me a couple days ago was sometimes our water, our cold water, infiltrates into our benthos, into the subsurface, and moves laterally along the ocean floor until eventually it pops up. And pops up in these outflows and these seafloor fluid discharges, which we are currently searching for. And it carries with them lots of chemical energy, which is utilized by our chemosynthetic bacteria to convert it into sugars that allows other organisms to tap into that energy source. So really, really important work by our colleagues who are looking at heat flow mapping. We're also going to try, as we see a beautiful anemone come into the picture with his corresponding squat lobster guarding him. Something we've come to expect now. Oh, we got some movement. Probably reacting to the ROV a little bit. Beautiful shot there. We would also continuing with our, I'm gonna stop talking a little bit and let's see what we see. Oh, there's a fish in the water column in front of us. He's moving, yeah. But there's something else coming out of the darkness. Pretty large crab. Can't tell you what species, so I'm just going to say a decapod. And he has no fear of us. Wow. Most likely a very thick, strong exoskeleton. As you may imagine, the pressures at this level are quite large. For every 10 meters down, we go up in 1 ATM. And incidentally, our ROV is rated for much greater depths than we are currently. We can go down to about 4,000 and a half meters. And you see the lights, the lasers on him, so we're able to measure his size. He looks to be about 30 centimeters across. That's pretty big for a crab, from point of the leg to the point of the leg. We tend to measure the carapace when we're doing fisheries studies, however. So nice guy there. So another one of our objectives are going to be identifying and characterizing benthic and pelagic sensitive marine ecosystems and biodiversity hotspots, including, as I've just mentioned, methane seeps, mesophotic and deep-sea corals and sponge habitats, and at several priority sites for Ocean Discovery League global deep-sea exploration goals. We're also going to be collecting samples and testing the feasibility of shipboard cryopreservations of benthic invertebrates for biodiversity preservation using a first-in-class cryopreservation medium. So some cutting-edge research. And on the screen there, we're seeing some human debris. It looks to be a bottle. Possibly a bottle. Plastic, yeah. Yeah, it did look like a bottle. I don't want to call the name of the bottle. I think it is. We're focusing strictly on taking of biological specimens. Octocoral and some sargassum. And for many of us, this is the first glimpse we've ever had of the deep sea. And it is definitely the first time I have had a glimpse of Trinidad's deep sea. So this is just the second site. We're going to be exploring all of our, well, as much of our EEZ as possible. We have multiple dives coming up in the following weeks. So don't forget to tune in every day. You may catch a glimpse of something no one has ever seen before. Another one of our objectives is characterizing water column structure and dynamics, as well as links to biodiversity, including phytoplankton, zooplankton, and even larval ichthyoplankton. Ichthyoplankton are larval fishes. So we'll be looking at some larval fishes, and we'll also be looking at analyzing their eDNA, which is a very exciting part of genetics, new part of genetics, where we can actually sample water and extract DNA from the water and see what sort of organisms were in the water. That's why some of our colleagues are focusing on sponges, which are filter feeders. These sponges filter the water, and they would filter DNA from many of the organisms that swim around them. So by sampling sponges, the tissue of sponges, we can not only get a sequence of the sponge, but we can also sequence many other creatures that live and inhabit these waters. So some really exciting lab work also going on. A very interesting objective that we're doing, as we see a glass sponge. With its associated anemones and squat lobster and some sargassum. And as we zoom up, crustacean, and as we get closer, maybe we'll see some other things. So some anemones, the glass sponge itself, the squat lobster. We actually see the air quotes roots, which are spicules that allow the sponge to adhere to the benthos. Those spiny looking things at the bottom, they're actually not roots, they're called spicules. And they help to anchor the sponge. Another interesting objective of this project, of this expedition, is evaluating the representativeness and connectivity of potential marine protected areas in Trinidad and the high seas. Currently, we only have one marine protected area in Trinidad. That's the Boca Reef Park. There is work being done on the northeast peninsula of Tobago to create a marine protected area there, although it hasn't entered into force as yet. And this information can help to inform those sorts of processes, because at the end of the day, decision makers require information. And that's what we're doing now. We're gathering the information to give to them, analyze and give to them, so they can make informed decisions in order to protect our wondrous biodiversity. So we did see some garbage bags, some plastic bags, some bottles. And why we focused on it is because another objective is assessing anthropogenic impacts from marine debris, microplastics, fisheries, chemical contaminants, oil and gas extraction, and climate change on the deep sea. So we want to see how these anthropogenic impacts affect our deep sea. have a sea star just chilling. And a soft coral off to our left. So our echinoderm, pentaradial symmetry, as you'll see, closely related to sea urchins as well, also contain that pentaradial symmetry. You'll see those 1, 2, 3, 4, 5 arms, characteristic of the family, as well as you can actually see some podia on the bottom of the organism, those little structures there. Tube feet, we call them. So there's an ambulacral groove underneath each arm, and it funnels food material towards the central mouth. Incidentally, these organisms have only one orifice. And keep the comments coming in on the chat. Whilst we can't get to all of them, we do try to read all of them. Ooh, what's this?
I think we can collect that.
Got a little, it looks like a snail.
It's like pearl. You see those? I like that snail. It's the third one we see.
Pardon?
It's like the third one we have seen.
So we can take it.
Kind of slurp it.
Excuse. We want to slurp this
I
one.
mean, we can try.
We could try.
It's seeing really fast.
So we're gonna try to take this one. We've seen quite a few of them, so our policy is if we see one, we're not gonna take it. But if we see multiple copies of them, we will try to take one for identification and for genetic research, as well as to house in the museum. So we're gonna use our suction device to try to slurp him up.
It's okay to use two?
Yeah.
Sorry?
Suction.
Yeah.
Yeah,
I mean,
two.
only one is full, so you can take number two.
Two's fine.
And then
I play a lot of PlayStation and I fly helicopters, and this is way more complicated than flying a helicopter. On PlayStation, that is.
Que eu apertei, mas é.
Thank you.
success. So we've successfully captured the snail. I mean, I saw a question in the chat. Are there a lot of buttons? There are over 100 buttons here. This is a really complicated process. You have not only to think about the buoyancy, but the thrust, and you also have to manipulate the arms in a medium that is not air. We're not accustomed to doing things here. So our ROV, I'm not blowing smoke here, our ROV pilots are some of the best in the world. And with that successful capture, my time with you is at an end. I'll now pass over to my colleague Charlotte.
Fancy here, going out with a bang with that one. Hi everyone, I'm Charlotte Tom. I'm from Trinidad and Tobago. My background is in environmental management, and I'm here on this expedition as a member of Species. Species is the lead organization on this landmark expedition. So whether you've been following along or just joined, I'll be taking you through this last hour of our second dive here on this Deep Wonders expedition. So what you're seeing right now is a screen of our ROV Sebastian, currently at depth 891 meters. So let's see if we find any more cool species for this last hour. So we will be stopping at many other locations throughout this expedition. I'm trying to do as many dives as possible with Sebastian. And so far we've already seen a lot of amazing species, most that I've never seen before actually.
Looking at the work. It's like my English sometimes goes away. Oh yeah, it's another one.
Thank you.
Very good expeditions. Free.
And look, we have a
Yeah, one is Schmidt.
halo fish there. We've been seeing many of those. Eel-like fish. We have a sea pen in the corner. Also seeing many of those octocorals. And an urchin. Another common find over the last 2 days. So as we move through the deep sea, what you're seeing here, these little white dots on the screen, is what we call sea snow. And this is basically made up of organic matter that is drifting all the way from the sea surface, made up of fecal matter, plankton, algae, and other organic matter. And this is actually like a major food source for a lot of deep sea creatures. So you'll constantly be seeing this sea snow as we explore through the deep sea with Sebastian. There we are, another halo fish.
You want the 4K? Okay, here. Yes.
I think I see this. See another. Oh, it's a halo. Couple shrimp swimming around there, also very common we've been seeing. If you've been watching for a while, you may notice these two dots on the screen, which are a point that help us measure the size of the organisms. And as you may have noticed, the deep sea has very small organisms oftentimes. There's very intense conditions in the deep sea. It's very cold, it's very dark, high pressure. So it's to its advantage to not evolve to be too massive and conserve energy with the limited food as well. See the sea star there. Also a common one we've been seeing over the past few days. Now everything that we've been observing as we dive through, whether we're zooming up on it or not, we've been logging it. So we have a team who's sitting behind me that's logging every single observation we see. So we can build up our repository of local data for Trinidad and Tobago's deep sea. And we've also been taking a few samples where possible. So if you were just tuned in with my colleagues here a few moments ago, we would have just collected a sea snail. And how it works is our ROV, Sebastian, has different bio boxes and different tools and storage units that he could use his arms to kind of pick up the samples and store them away and bring them safely back to the surface. What is this we have here? Let's see if we could take a closer look. Here we have two Venus trap anemones. We've been seeing quite a few of these. Very much resembling, of course, the terrestrial plant, the Venus trap. And if they are disturbed, they will close up in a similar way. Lots of sediment and particles rising here. Hopefully it settles and we can get a closer look. Right here we can see a sea urchin, and in the distance, what is that?
That's armored sea robin. Armored sea robin. Armored, yeah. Sea robin. Robin.
Here we have a confirmed with our one of our lead scientists here, an armored sea robin. And as you may notice as well, a lot of the deep sea creatures we've been finding, for example, the shrimp or the sea urchin, have a sort of red-like color, and that's because red is the first color to kind of disappear as the sunlight enters the water. So for the deep sea, that becomes sort of like a camouflage, almost like wearing a black sweater in the nighttime. Here we have more fish in the distance. As you would notice too, these halosaur fish that we've been seeing frequently have more of an eel-like tail rather than the shallow water fish that we might often see. And this is also a evolutionary purpose to conserve energy and sort of just glide through the water more easily. I see another shrimp in the distance there, swimming away.
So we'll get like it snaps. So that's
It looks like plastic as well. A bit of
swimming on that.
big patch of sargassum, and it seems it's possibly some plastic wrapped up in there as well.
No, brilliant.
Yeah, so even from the first dive we did yesterday, we've been seeing quite a bit of plastic remnants here and there. So you can see it made its way all the way down to the seafloor. About 1,000, close to 1,000 meters down. What do we have here? It looks like it might be another tripod fish. I think we've been lucky enough to see about 4 or maybe even more of these today. And I believe we also saw one yesterday.
drop some C4 right there, and then next to the time I come up. Because when you on the week,
Yeah, we can keep going. Whatever, if it's a trash, we can stop.
trying to make a clean.
We're gonna keep on going, see what else we can find. We see another shrimp and sea urchin, as well as a sea star. I think that's a sea sponge down at the bottom. Coming up here, we have another sea pen, which is an octocoral. And it looks like it has a friend laying on it. Now we're looking at another hollow sawfish and a shrimp just floating across the water there. As you notice, we're also seeing a good bit of bits and pieces of sargassum that's made its way all the way to the deep sea, which we typically see on our beaches and floating in patches on the sea, but it's made its way all the way down. And what we just went past there, the little palm tree looking like structure, was a crinoid. We got a sample of them earlier. And right now we're seeing, it looks like it's a can. Yeah, crushed up can has made its way all the way to the bottom. So a bit of behind the scenes. So what's going on right now? We have a whole team behind here, kind of piloting the ROV. A very skilled team, and we also have our lead scientists in the room, as well as some loggers helping us identify everything we see and ensure that we log it accurately. As we're now in the last hour of this dive, soon or already we'll be prepping for when ROV Sebastian returns to the surface. And we have to very carefully handle all of the samples, get good pictures of all of them, so that they could then be off to be preserved as well. If you see here coming up on the left, we have a glass sponge. It has that name because it has a skeleton made up of glass-like structures. We got a really beautiful sample of one of those yesterday. We also have an urchin on the right of the screen there, and some more sargassum. And just floating at the top of the screen there, we have a comb jelly. Let's see if we can get a closer look. Wow. Very mesmerizing to look at.
take much.
So already in the past 2 days, we've seen some really amazing species, and a recently published study by my good friend Jamie Luchin, also in the room here on the expedition, documented 451 species already observed in the deep sea, Trinidad's deep sea, but estimates that the actual number is about over 1,600, meaning that more than 70% of deep sea species here in Trinidad and Tobago remain to be discovered. So hopefully we can see some more new species in the next few dives as well. So the handful of deep sea scientific expeditions conducted so far have been mainly undertaken by foreign researchers who have left very little data or samples or understanding in country in Trinidad and Tobago. So that just re-emphasizes what a landmark this is for us to be a Trini-led team, the first time ever leading this Deep Wonders expedition right here in Trinidad and Tobago. So everything you're seeing on this dive stream is never before scientifically explored parts of our deep sea. Oh. Wow, see another crab here in the distance. Pretty big one, it looks like. Let's see if we can get close to him without scaring him away too much. Oh, wow. Slowly moving away.
Great if you come to our port.
See another fish here on the right hand side. Very common. There we could see Tino Fall. Very beautiful. What's this in the corner here?
It's a spider.
Spider. In the bottom right there, we're seeing a sea spider. Very fine legs sticking out. This might be the first one we're seeing today.
It's a spider. I think you have to get whatever. It's a spider. Seems like a dead animal. Make sure to take really good zooms of this spider.
See if we can get some nice close-ups of the sea spider. Wow. Oh. And another fish right in the background. couple more actually.
This time, alright. Yeah, it might be like a slower change.
What we're looking at here, we think, is a shell, possibly of a sea urchin, surrounded by some more sargassum. Let's take a closer look at this guy. Looks like might be a squat lobster. Is that a squat lobster? Oh,
Like a tiny one.
tiny squat lobster.
I know the difference between squat lobster.
We've been seeing a lot of
In
these.
Spanish, we only say peje. But I think it's a six. Everything is a squat lobster. The tiny one.
Again, zooming back out, we see our sea spider friend here and some shrimp
Oh yeah, the opioid.
as well on the left hand
This,
side.
Then the tiny arm. So it's like a breed or something.
In the distance, coming up, it looks like there's a glass sponge.
Sorry?
Glass sponge.
Yeah, it is.
Glass sponge.
Are we only movement?
These are very fragile. got a sample of them just yesterday. It's a very intricate pattern on it. Let's see if we could zoom in and take a closer look.
Sorry?
Oh,
What it is?
could we zoom out to the
That one looks a lot different from the one we
I
collected
know,
yesterday.
that's a CRT. I think it's a CRT. That's different.
Looks different. This first one we saw, right?
We want to try to take a piece of this one, not the whole thing.
Just a piece.
Okay.
Yeah, that's what this
So we
says.
should fire Niskin first.
Yep.
Okay.
So Niskin first, as close as possible without touching it.
Like first we will fire one of the Niskins. So it's the water samples.
Mm-hmm.
It's the first one we take
It's a
down.
new
Then we will try to take out just a tiny piece sponge and put it in
Yes,
a correct
that's
river.
right.
So correct river soup, so it's
Okay. We do one of the skin muscles and then try to get to core equivalent. Okay. We're gonna wait on this to clear up a bit and see if we could actually get a sample of.
so they will fire two Niskins first. They will try to clear us.
And then
But you will see which number it is. We should wait.
You think we could find another one like this?
It's the first one we see.
This one?
Yes. So that will be. Do you think it will take a lot of time for the sediment to settle up? Yes, that's it.
You know where is it, man?
Yeah, it's just where we start to move
Just
back.
come on together. Because more you try to stay above, because the sub is heavy, it's continuously pushing. You just stay up.
So right now we're hoping some of the sediment can clear up so we can get a sample of the sponge we just saw, which is the first that we've seen on this expedition so far.
Then come.
So right now we're on our second dive of the expedition, and it's slowly coming to an end. Just about a few minutes left till we try to ascend, but we're gonna see if we can get these samples first. As it slowly starts to clear up, we can see a little red shrimp, deep sea shrimp in the back. Oh, and if we look closely, we can see a jelly slowly floating up across the screen.
Yeah, yeah.
So we always take pictures, so it's better if we go.
I take picture.
We want to take pictures.
Yeah.
Forward to where you were.
It's like
You guys just have a few open. I mean, you probably just. What time you guys want to? Yeah, so I would have just do the next 5 minutes if you see one. Okay. Go ahead. I really moved
Repeat your message, please.
it. Yeah, back on the move.
Back on the move, okay.
I mean, it's really hard. We will
How
not
about
be
this
able
one?
to get rid of that
as the sediment slowly starts to clear up, gain visibility again. We can see a sea pen here on the right. We see another taller one in the distance as well.
We are in the middle in 5 minutes. So.
Do you know what you're doing wrong? Why straighten up that bit?
Unfortunately, we couldn't find back our sponge, but hopefully we see many more on the dives,
Sorry?
where we have a few more minutes left of this dive, and then we will start our ascent. Thank you everyone who's been following along. This is
I mean, sometimes
the first time we're seeing some of these parts of our Trinidad and Tobago waters, and there are people tuning in from all over. So thanks for following along. I'm sure we'll see lots more exciting stuff.
No, that was actually my so,
This is only our second dive, and we're hoping to do many more as we go to the many different sites that we'll be exploring.
yeah, we're
We see another sea sponge, fish, and a shrimp rolling around.
We can start closing.
So once again, everyone, just want to say thank you all for tuning in for this second dive of our Deep Wonders expedition, the first of its kind, Trini-led expedition here in Trinidad and Tobago's deep sea. And we'll be doing many more dives, so be sure to tune in. Thank you all. I'll hand you over to the ROV pilots.
That's it, right?
Yes.
Bridge. That's our V of bottom.
Sorry, yeah, it's a more assembly.
Bridge.
The ROV of the
Do
bottom.
it.
Roger.
Like a general. If you want.
Yeah?
Yeah. I mean, that we also have that in the report. It's like not in, but good.
You sure?
Do it.